To introduce myself, I'm Sambit Sahu. I am a senior bioinformatics scientist at Mission Bio, and the agenda for today is I'll give an overview of the Tapestry R package, what it does, and the visualization you can produce, and what data you can explore. And major focus will be a hands-on activity. And I believe in the morning there was some technical difficulty with the hands-on, so please feel free to ask more questions during this webinar. Um, to get started, where you can download the package, it is at portal.missionbio.com. We'll go there in a bit, and we would need some data sets to get started. This is at this link, portal.missionbio.com/datasets. And there's a bunch of vignettes and documentation available inside the package for the functions and methods that are used. And any further support, you could contact your local affairs. So the Tapestry R package is an exploratory data analysis tool which you use at the end of Tapestry pipeline results generated, so that you can start with the Loom file or the multi-omics protein and DNA files, and you start analyzing your data. You could cluster your cells based on genotype and look at protein expressions. You could visualize protein levels on genotype clusters. You could look at copy number variation and combine it with the protein data. And overall, you could look at this multimodal profiling of your single cell data, which we are going to look in a bit. So this is a bit rich slide, but I want to emphasize the three outputs which are coming out of the Tapestry pipeline and how we process each analyte separately in the R package. So the first output of the pipeline is the genotype data, which is your per cell is the mutation, is a heterozygous, homozygous, or wild type. You could load that data. You could filter the data based on some basic filtering we have. You could cluster those cells based on your SNV and indel, and you could visualize the data in a scatter plot or heat map and gain further insights. The second data set we have is raw reads, meaning for the Tapestry data, you have per cell, per amplicon, how many reads are mapped. And you could use that data to first normalize by some methods we'll go over, and then you could define your diploid cells. And you could estimate the ploidy of the tumor population of cells based on that. And you could do a bunch of clustering and visualize SNV and CNV data together in a single cell. By some methods we'll go over, and then you could define your diploid cells. Hey, Joan, I think we are hearing an echo from your side. Sorry about that. Then the third is our protein data, which is the output of the Tapestry protein pipeline. You get per cell, per antibody tag, how many reads went, and you initially keep the cells you call from DNA. We would normalize the counts matrix. We would cluster the protein data, and then we could look at similar visualization. The second way is to merge all of these three analytes and how we can look at it, look at this multimodal profile together. So this is our protein data, this is our genotype data, this is our reads matrix, and we merge using the Tapestry package. It creates something called a multi-assay or multi-analyte object, and we would cluster it, and we could define cell clusters based on genotypes. And then we could either define cell cluster based on protein and look at it together. There's a bunch of visualization you could do. We could look at in scatter plots, SNV, protein heat map, correlation plots, and then we could overlay each analyte on top of the other and look at it. And then we have a ridge plot way of visualizing the data. This is a small example of how the results look like. The first plot on the left you see is just based on your mutation data, how the cells are clustered. I'll go over the data set in a bit. So this was a 4-cell line mixture, and you could see how 4 cell lines are clustered. Similarly, for the protein, you have for the protein, and just to remind, these are UMAP axis here. And you could also cluster based on SNV and protein together in the third plot. Now you have the base projection, which is, let's say, protein, and I would want to look how are my antibody counts expressing. So we can see for CD3 in this cluster is highly expressed, CD90 is highly expressed in cluster 3, and this is how we can look at these two data sets together. Another visualization is regarding ploidy. So for the same 4-cell line mixture, and these are the gene we could look at on the copy number gain and loss based on the diploid cells. And then you can also look at in line plot how which genes were elevated or lost. As I mentioned, you can look both protein and SNV together in a heat map. So this is one clinical sample where we looked at different population. You can see that the populations defined by a SNP has a differentiating characteristic in protein layer also. We could look at SNV and protein correlation. Each y-axis here would be SNPs and then the protein. And we can say also in a ridge plot, these are SNV clones defined by mutations, and then we can show how each of these proteins express. So without further ado, let's get on to the hands-on part. We'll go to this link, portal.missionbio.com. It will ask you to log in, and you can also sign up for free to get this package and the data sets. So in the bottom, you will see this tapestrR package. You can just download it, and then there will be a data sets tab there. We are gonna use just the first data set called 4-cell line mixture. If you click on that, it'll give you a little bit of summary of what this run was. So as I mentioned, this was a 4-cell line mixture, Raji, K562, Tom1, and KG1 run on Tapestri Platform. 1/4 of the Tapestri run was sequenced on an NextSeq experiment. Tapestri produces 8 channels or tubes worth of cells, and only 1/4 of that. That means 2 tubes were used to process this. And we had around 1,200 cells. So okay. So this markdown file and the vignettes are also available in the package. I have downloaded the R package here, and the data set, if you unzip, all the files which are needed for this analysis are in that data set. So first, I'll set my working directory where all the data is, and then you would install that tapestrR package, which is a zip file. I have already installed it, so I'll just load the library. So we would need 4 files from the DNA pipeline, which are my amplicon.bet file. We need the redistribution TSV file. We need the loom file, which contains all the genotype and quality information. We would need the VCF header file, which is all of these are in this 4-cell line multi-omics zip. And then you would need the protein data, which is output of the protein pipeline, and it's still in the same zip file, which is the adtcounts.tsv. So what we can do is quickly load this data set. First, we'll load the barcode file. You can even look at what every function does. So what does the read barcodes do? It reads the distribution file. So we're gonna connect to the loom file, which is the genotype information. And then we will load the loom file. And there's many filtering options based on GQ, allele frequency, and which are usually the same filters we have in Insights. And you can use the same filters to filter your variants and cells. So after you have the filtering done, we will convert that SNV to a SNV object. This convert to analyte will convert the data to an SNV object. Yes, there was a question. Yeah, this is, I think, 3.6 version. Okay, so now if I look at my SNV1 object, it will tell me I have 1239 cells. I have 52 features after all the filtering. And features are my positions. And if you look at and this matrix and the data will contain all the cells and all the mutations information. And 0 would mean wild type, 1 would mean heterozygous, 2 would mean homozygous call. Okay, and I'll just assign a name for that. And now I will read the protein file, which is this read protein count. Again, you can look what this function does by doing a question mark. Create protein analyte object. And if I look at the length, these are all the barcodes which were coming out of the protein pipeline. I have not done intersected with the DNA cells yet. Okay, and this intersects with the DNA cells, and now we have the same number of barcode with DNA. Next is read DNA counts. So this actually creates the read counts object, which we'll be using for copy number estimation. Create DNA read count object. And we'll have, there's some differences, but when we create a multi-analyte object, all of these will be similar. Next, we would want to normalize the data for the protein data. We support multiple methods. Here, I'm going to show the CLR normalization, but there's many normalization methods with mean, median, CLR, and some you can look at in the vignette. So I'll normalize the protein data here. And the DNA1 object, which is my copy number or read counts data. Multiple normalization methods are supported. I'll use the median normalization here. And this has 121 features. We have 1241 cells here. The next step, as I mentioned, we're going to combine all of these 3 analytes into a single object, which is done by the merge assays function. The merge assay will will merge multiple analytes to create a single object. Now, if I look at it, you tell me there's total 1236 intersected cells. In my SNV, I have 52 features. In the DNA read count assay, I have 121 features, and in protein, I have 10 features. Now I can just explore the matrix. How does the data look like? So this is the SNV data set, as I said, for each barcode, for each variant, we can look if it's a wild type. 1 would mean it is heterozygous. 2 would mean it is homozygous, and then 3 would mean no call. Same for protein data. Okay, after, so now in this state, yeah.
There are just a couple of questions coming in through just on versioning. Do you mind taking a look at those?
This is R Studio version, and I think it is 3.6 version. Version 1.2, so R Studio, and R version is 3.6. Okay.
Thank you.
So I have my SNV and protein data set. I just created a SNV plus protein data set here by combining the data sets. We would want to estimate the number of clusters first. So what we'll do is we'll need to find an optimal number of clusters, and we do that by silhouette analysis. I'm gonna show that. So this was a 4-cell line mixture, and by silhouette analysis, we can see there are 4 clusters coming. And we're gonna use that as an input to k-means when we cluster the genotype data. This is the dimensional reduction method, which would use, we use here UMAP, and if we could also do t-SNE. And for SNV, we use a Hamming distance approach for this UMAP projection. And then let's see how it looks. So this is the genotype data, and we see 4 clusters. Next, we are gonna move on to protein data, and we're gonna do similarly dimensional reduction using UMAP. And you can use any projection like t-SNE. And similarly in the protein space, since these cell lines have distinct antibody tags, you can see these 4 clusters. You could also do protein plus SNV together. Let's see. What's the percentage of dropouts, unknown genotype do you have in this data set? That's a good question. So the filtering which we use above, that actually get rid of the cells which have large number of mutation which are no call. And you could play around with your filter if you want to be more stringent and want cells which are more complete, in the sense all of my required mutations are there, in the sense all of the amplicons have reads. You could use that filter. So for this data set, based on the filtering, that would change what is being processed in downstream.
Just a question from the audience. What percentage of dropouts or unknown genotypes do we have in this data set? Do you have that available?
I just answered that.
Right.
Yeah, I just answered that. So based on the filtering, that would change here, as I mentioned. If you want 50% of cells to have, if you want all the cells to have more than 50% variants to be genotyped, then that would be my threshold, and that is the filter called, I go up, And these are all documented. That's the filter called GTM, so minimum variance. So this is a percentage. I would increase that to 50.
Yeah, and there was another question on the Q&A. On top of the number of clusters, when you compare clustering patterns on different samples, does a different location of clusters of the same color cluster have any biological meaning?
Yes, let's get to that. After I have combined, that will complete the biological story.
Okay, thank you.
Hold on for a sec. And I think at the end we'll open up for a complete question. Let me finish this up. You could cluster this up based on both SNV and protein. I guess it's taking a bit longer. I have these things run. So um, let's look at one of the stories. So based on based on our bulk sequencing, we know that there are two mutations, TET2 and EZH2 are homozygous for Raji cell line. And I would want to look at this together. So if I look at these two protein markers, CD45 and CD19, and also TET2 and EZH2. So these are homozygous, and also these are high for my CD19 and CD45, which are also the protein truth for Raji. So and this is also the cluster, which we can look at the same data sets in the scatter plot. So what I'm doing is we have those projections done on protein, and here I will just say that which of these clusters are positive for EZH2 and TET2. And this is the cluster which is positive for both. That means homozygous for both of this mutation. And it looks like CD19 is high for this, which tells you based on this cell line experiment, your truth would correlate between mutation and protein. So I would stop here and open it up for more questions. Okay. How do you decide which normalization method to use when performing normalization of each data slot? So for a protein dataset, usually we use CLR, which is centralized log ratio. You divide the reads by a geometric mean. And for mutation, for read count data, we usually use median or mean for normalization. That is based on, you have to try it out and see which method works best for your data set. That's why this is an exploratory tool. We provide multiple normalization and clustering methods, and yeah, you have to try it out. In the UMAP produced by SNV data, are the separate clusters equivalent of different genetic subclones? Yes. So I have not shown the truth, but I have one figure. So we know that for Raji, we have if you take this 4 mutation, we have different genetic signatures for each cell line. So I just showed these two TET2 and EZH2 mutation, which are homozygous, and which shows the different genetic signature. I think yes, this tutorial is being recorded, and we'll have a YouTube link for this.
Sandeep, there are some from the Q&A as well.
Okay.
So one question, I don't know if you can see them, but I'll just go ahead and ask you. What is the total number of proteins that can be detected per cell with the current Tapestry system?
Total number of protein. So I think we have tested around 30 antibody tags. Depending on your system, we would have to design those panels.
Okay, great. Yeah, and I know just in terms of the product that we will be launching a little bit later this year with BioLegend, we plan on, I thought that the number was upwards of 40, but we will get back to you as we become closer to launching that with BioLegend.
Yeah, and your local affairs would be the best point of contact.
Where can we get all the codes for practice? Do you want to give them the link there?
Yes, so the R package is available on portal.missionbio.com. You can download the zip file, and if you open it up, all the R code is there, including this markdown file and this notebook which I was running. Everything is in that package, and just to download the data set with it, and you can get started. And if you have any question, just shoot us a message.
A couple more here on the chat. Will this, and I can answer this one, will this tutorial be recorded and uploaded on YouTube? In fact, it will be, and it is right now. We are actually streaming live on YouTube currently. This will be on the Mission Bio YouTube channel. So at the end of this webinar, you should be able to access that recording on our YouTube live channel.
Yep.
And we will also follow up with an email to everybody with the appropriate links as well.
Yep.
Another one, if I do not have genetic pre-knowledge like you do for ECH2 and TET2, for example, what can I do?
Yes, that's why we provide this way to estimate the total number of clusters, where we do silhouette analysis and tell you that there's going to be 4 clusters. And now let's say you have a clinical sample and you have no idea of the genetic signatures, and you would go and look at what mutations are prevalent in that cluster. And that would be a starting point. And then if you overlay your protein signatures, you would have better idea coming from both sides, what is the characteristics of those subclones or clusters.
Right.
Can your pipeline now handle serial specimens, in particular to clonal evolution throughout therapy? That's a great question. As I said, we can merge multiple analytes, multiple assays. You could also merge multiple sample. So there is a function which is merge samples, where if you have one sample, you have DNA and protein, and you did a drug treatment, you have another sample, DNA and protein, you could merge those 2 samples. Those will be tagged as time point 1, time point 2, and you can do the same visualization on both the analytes using this package.
Sambi, do you want to talk a little bit about how Insights can do sort of like fish plotting and longitudinal samples that way?
Yeah, so inside that is for just DNA analysis, where if you have longitudinal samples where your clones are evolving over time, you could use insights where you look at longitudinal samples, how the mutations changed and how the allele frequencies change across different time points. Sort of phylogeny, giving you an idea about phylogeny.
And then they can download Insights through the portal as well. Is that correct?
Yes, same portal has the software Insights, which the input to that is a Loom file, and it is readily out there. Can I use Tapestry to detect unique protein signatures from multiple clinical samples? I guess you mean you have multiple samples, and we would want to look at the protein signatures. Yes, you can. Using the same package, if you have multiple samples with just protein analyte, you can merge those samples and look at it.
Okay.
Best
Any other further questions?
Yeah, there's one question I think that we can take at the last. Are there any best practices, recommendations for sample batch effect correction and or modeling specifically for the scDNA read? Sample batch effect correction, we have, you can look at multiple samples together in Insights, as Hannah mentioned. That's the software you can download directly. And the default filtering should get you to those answers you're looking for. And and then for the copy number part, if you're asking about single cell DNA reads, which you would ideally use for copy number estimation, the normalization within sample and across sample would be helpful. And for your mutations, you can also use a blacklist file where you do not want some mutations to come up in your analysis.
Great.
Okay.
Okay.
Yeah.
If no further questions, then we will conclude today's webinar. We appreciate all of you for joining us. And as we said, we will have the recording on our YouTube channel if you'd like to refer to it there. And we will also be following up with you with a link to the recording if you miss it on YouTube. And then just to let you know, for next week, we will be having additional scientists from the Mission Bio team just speak a little bit more of a higher level overview of why one might want to do single cell. So we encourage you to join us for those sessions, and those links for registration will be available shortly. And then also if you are an existing customer or just interested in spending some time with our field application scientists, we will be hosting office hours on Thursday at the same starting time. Anyway, we thank you all for joining, and we hope that you stay safe and healthy.
Thank you.
Thank you, everyone. Bye-bye.